Next, we're going to talk about how neurons send and receive information. Anytime you send and receive information, you need some kind of medium. For example, when you're using the old-fashioned landline telephone, the fixed telephone with a copper wire connected with each other, you are using electric signals to encode the information and send from one place to another. Nowadays, most people are using cell phones. With cell phones, you don't use electricity as a medium. There's no wire, it's wireless. You use some kind of waves to transmit the information from one caller to another caller. Neurons also need media in order to transmit information. Neurons use two types of media. One type is electric signals, electric currents. Another type of a media is chemical agents, what we call neurotransmitters. These two processes happen in different parts of the neuron. Let's look at how neurons send information to other neurons. This happens in the axon. Axon is a part of a neuron that is responsible for sending information to other neurons. Here I have a schematic representation of a neuron. Here you have the cell body, the soma. I'm missing the dendrites. I don't have any dendrites in the picture. And here, this long extension is an axon. Axon uses electric currents to send information. How does an axon generate electricity? Axon, when sending information, it has three different states. The first state we're interested in is called resting state. When the neuron is not firing, when the neuron is not sending or receiving any information, when it is resting, when it is inactive, you can see there's a difference between the two sides of the membrane. This is the axon, and outside the axon, outside the membrane, you have a more positive ions. These are positively charged ions. Most of them are sodium ions. Inside the axon, inside the membrane, you have negatively charged ions. There's an imbalance between the two sides of the membrane. This is called depolarized. It's like a battery. If you look at any AA battery, it has a positive end and a negative end. This is called polarization. When the neuron is not firing, it is polarized, with positive ions outside and negative ions inside. This is not a stable state, unstable, because positive ions and negative ions attract each other. They try to get together. There's always a pressure for them to get together. The reason that in the resting state, ions, axon can remain polarized is when in the resting state, the membrane is not porous. The gates, there are little gates, pores on the membrane. In the resting state, those pores or gates are close to positive ions. So the positive ions have to stay outside. They can't get in. when the neuron fires, when the neurons are sending a signal, the pores or the gates on the membrane opens to positive ions. And the gates don't open all at once. It's progressive, starting from the base of the axon, starting from close to the cell body, and then progressively open to the tip of the axon. This way, electricity gets generated. When positive ion gets into the negative, get into the axon, it gets mixed up with negative ions. When positive meets negative, there's electricity. This is how electric current is generated and transmitted inside the axon. This current is called action potential. And it's also called impulse, electric impulse. And after firing, after a neuron fires, before it fires again, this is the third state. This is a refractory period. So before the neuron can fire again, it has to become polarized again. It needs some time to recover the polarized state. It has to move the positive ions that has gone inside out again. So you have a slightly more positive ions outside the membrane than inside the membrane. You return to the polarized state. This takes some time. And before this process is completed, the neuron stays inactive. It cannot fire again. This period is called refractory period. Refractory means no action, it's inactive. This is how electricity gets generated in the axon. And electric current or action potential or the electric impulse is the medium that the axon uses to send information to other neurons. There are two kinds of connections between neurons. One kind of connection is called facilitative. Another type of connection is called inhibitive. This meter shows facilitative connection. You have two neurons, neuron B and neuron A. Neuron A is sending stimulation to neuron B. And for facilitative connection, when neuron A fires, neuron B fires faster. For an inhibitive connection, when neuron A fires, neuron B fires slower, slows down. This meter shows a facilitative connection. You are recording with this meter electric activities in the receiving neuron. This neuron is receiving stimulus or stimulation from another neuron. When there's no stimulation from the other neuron, this neuron fires at a random rate. Neuron always fires even without stimulation. It fires randomly when unstimulated. But when it receives a small stimulation from the other neuron, it increases its speed of firing. When it receives a larger stimulation, it fires even faster. So this is a facilitative connection. Inhibitive connection works just the opposite way. When one neuron fires, the connected neuron, the other neuron slows down. This picture shows a neuron. This long extension is an axon. A neuron has a large number of dendrites. remember dendrites receive information from other neurons. Some of these dendrites receive facilitative information. Other neurons are trying to urge you to fire faster. Other dendrites receive inhibitive stimulation. Other neurons are urging it to slow down. Sometimes a neuron receives a conflicting stimulation from different dendrites. How would a neuron make a decision how to resolve these conflicts? A neuron is like a computer, like a little calculator. It computes average. If on average, it receives more facilitative stimulations, it increases firing. If on average, overall, it receives more inhibitive stimulation, it slows down its firing. Neurons are quite smart. Because axons generate electricity, we can monitor the electric activities in the brain with a device called EEG, electroencephalograph. And we can record the electric activity in the axons in different parts of the brain. EEG can be simple and can be complex. Simple EEG have just a few electrodes. These electrodes are attached to the scalp, outside the scalp, and then it can record electric activity beneath that part of the scalp. With a complex EEG, you can have many electrodes, 30, 60, even more than 100 electrodes. Of course, with more electrodes, you can cover a greater area of the brain. The machine records electric activities in the brain, and you can see the brain's electric activity shows different waveforms, has different amplitudes, has different frequencies. While scientists know when people are sleeping, they show a particular kind of a brain wave, different than when people are awake. And when we talk about states of consciousness, we talk about different sleep stages. Stage 1 sleep, stage 2, stage 3 and 4, deep sleep, shallow sleep, rapid eye movement sleep. Each sleep stage have a particular characteristic waveform. For example, in deep sleep, usually people show this delta wave. We will talk more about brain waves during different sleep stages. Another way of using EEG to investigate brain function is to ask people to do something, to perform a task, or look at a particular stimulus, or generate a particular feeling, and then we record which part of the brain is really active, is sending a lot of electric signals. For example, well, we can make people view a calming scene or a scary scene, and then record which part of the brain is very active in sending electric signals. This way we can understand the different functions of different parts of the brain. Maybe we can find out which part of the brain is really active when people are calm, which part of the brain is more active when people are scared or when people are nervous. EEG is a very useful way, is very useful methodology to study brain functions. Now let's move down the information transmission chain. information is generated, electricity is generated in the axon, and then electricity travels down the axon to the very tip, to the very end, to the terminal of the axon. And the terminal and axon gets connected with dendrites of the next neuron, but this connection is not complete. There's no physical contact, but there's a gap between the axon and the dendrites. This gap is called a synapses. How does information cross the synapses? Can electricity cross the gap? No, electricity doesn't cross any gaps. If you cut your electric cable, you are out of power. Electricity doesn't jump across any gaps. There must be continuous connection, but we do have a gap. The synapses is a gap. Electricity doesn't cross it. What do? We need another signal. We need another messenger. Now, neurons use neurotransmitters. This is chemical stuff to carry the information across the synapses. This picture shows two neurons. This big neuron is sending information to this one. This neuron is only shown a small part. Here we have the nucleus, the dendrites. The dendrites receive information from other neurons. And then we have this long axon. This axon has a myelin sheath around it, so the information in the axon travels relatively fast. The electric currents or the action potential reach the tip, the terminals get stopped here. And now we need to mobilize the neurotransmitters. Neurotransmitters gets released from the terminal of the first axon, and then by diffusion, they swim across the synapses. And on the other end, on the other side of the synapses, you have receptors. Receptor sites are located on the surface of the dendrites of the second neuron, on the neuron that is receiving the information. When the neurotransmitter gets bound to the receptor sites, it stimulates the second neuron. The information gets carried across. So the second neuron fires in its axon. The second neuron becomes stimulated and its axon produces electric current. This is how information gets across the synapses. This is really cool animation. If you look at the top, these two neurons, they only show axons. Axon in one neuron, axon in the second neuron. They both rely on electricity, action potentials. They fire. And here in the lower part of the graph, you have neurotransmitters. Neurotransmitters get released in the synapses, cross over, stimulate the receptors, and the second neuron fires. So a neuron uses two kinds of messengers or two kinds of media to transfer information, send information, and receive information. The axons use electric currents, action potential. And across the synapses, the neurons rely on neurotransmitters. Another trick that happens in crossing the synapses is reuptake. Look at these two animations. The animation on the left shows a release of neurotransmitters. The sending neuron is releasing neurotransmitters, and some of the neurotransmitters get across the synapses and get bound to the receptors. But you see, some of the receptors are free floating. they have not bound to any receptors. These surplus transmitters, they get taken back to the sending neuron. Neurotransmitters seem to be very precious. You don't want to waste any of it. So you are releasing neurotransmitters in the synapses, and you are taking them back at the same time to recycle any surplus, any neurotransmitters that are still free to be recycled, to be used again. Suppose if the reuptake is very fast, then the sending neuron is recovering or retrieving the neurotransmitters back from the synapses really fast. This way, the neurotransmitters have very little time to stimulate the receptors. They have a little time to cross over the synapses and get access to the receptors on the other side. This would be like going to a party. If you want to socialize, if you want to make friends, you go to a party. But if you stay very briefly, if you leave the party right away, you don't have opportunities to meet new people, to make friends. Then, well, you don't have a social life. In order to really have the opportunity to meet someone you're interested in, you have to stay there. This principle applies to reuptake too. If reuptake is really fast, it's like leaving the scene really fast. You don't have a lot of opportunity to stimulate the receptor. In order to really give a strong stimulation to the receptors and send a strong message to the second neuron, really stimulate it strongly, the neurotransmitters have to stay in the synapses longer time. In other words, you have to slow down the reuptake process to send a strong messenger, to send a strong stimulation to the second neuron. This principle is very important in developing medications for some mental illnesses like depression. 3 factors determine how strong the first neuron stimulates the second neuron. Number 1, the number of transmitters released by the sending neuron, the neuron sending the information. If the first neuron releases a flood of neurotransmitters, and then the second neuron receives a strong stimulation. Number 2, it's the neurotransmitters. If there are more the second process, the second factor determines how strong the message or stimulation lies with the receptors. If you have more receptor sites on the dendrites of the receiving neuron, the message or the stimulation is going to be stronger. The third factor that determines how strong a stimulation is reuptake rate. If the reuptake rate is really fast, then the stimulation is going to be weak. If the reuptake is very slow, the stimulation from the first neuron to the second neuron is going to be stronger. This animation shows neurotransmitters being released by the axon, and then by diffusion, it crosses over to the dendrites of the second neuron to deliver the message, to deliver the stimulation. The second animation shows a reuptake. Any free-floating neurotransmitters is taken back into the second neuron to be recycled. One more characteristic of a neuron firing, the electric activity in the axons, a neuron either fires or not fire. A neuron doesn't have a strong firing or a weak firing. A neuron's firing is like a light switch with two positions, either on or off. You can't adjust how strong the light beam is. You can't adjust how strong the firing is. And then how would neurons reflect strong stimulation versus weak stimulation? When a neuron receives a strong stimulation, it fires faster. If it's facilitative connection, it fires more rapidly when the stimulation is strong. But when the stimulation is weaker, it fires less fast. Summarize a little bit, 3 factors influence the rate of firing of a neuron. Number 1, the amount of neurotransmitters released from the sending neuron. The more transmitters released, the stronger the firing, the faster the firing, given it is a facilitative connection. The second factor is amount of receptor sites available for binding the neurotransmitters on the receiving neuron, on the dendrites. Then the third factor is the rate of reuptake. How fast the sending neuron is taking the released neurotransmitters back for recycling. The faster the reuptake, the slower the firing in the next neuron, the weaker the stimulation. We know neurotransmitters are responsible for carrying information from the axon of the sending neuron to the dendrites of the receiving neuron. And there are different types of neurotransmitters. Some neurotransmitters we understand more. We know more about them, and we know their functions. Several neurotransmitters must be memorized in this course. One is called serotonin. Serotonin, you can see serotonin works in certain brain areas, and serotonin is responsible for mood regulation, regulating mood. This is one of the serotonin functions. It produces euphoria and depression. If a person has a lot of serotonin activities in the brain, the person usually experiences euphoria. It's a great happiness, pleasure. Sometimes you can call it a high. You get a high from serotonin. And if a person has a very little serotonin activities, really weak serotonin activities, the person may experience a depression, negative emotions. There's research showing that people with depressive disorders tend to have weaker serotonin activities. Certain drugs stimulate serotonin producing neurons and increase the serotonin activities. For example, the drug ecstasy stimulates serotonin neurotransmitters. So people get a high from using it. And now you may think that why don't we use ecstasy to treat people who have depressive disorders? Since depressed people have weak serotonin activities, then ecstasy seems to be a good remedy. Ecstasy gives them a boost in serotonin production. It may work for a while. It may work in the short term. Some depressed people do use ecstasy and other drugs to boost their mood, to make them feel better, to ease the suffering. But there's a downside, dependence. Well, the brain is recording, if the neurons rely on ecstasy to produce neurotransmitter serotonin, and then when it stops, when the person stops taking the medication, the brain, the neurons gets lazy. It produces even fewer neurotransmitters, serotonin. So the person gets even more depressed. And then the person must increase the dosage, take more ecstasy. This would become a dependence effect. This will become a drug dependence because of the withdrawal effects. So ecstasy is no solution. We have to find other ways to help people with depression. And usually, the medication for treating depression is by way of reducing reuptake. Look at here. Suppose this is the neural synapses in the brain of a depressed person. The sending neuron, the neuron sending the information, is releasing neurotransmitters, serotonin. Serotonin, these golden little bubbles, gets released in the synapses. Some of the neurotransmitters cross over and get bound to the receptor site to stimulate the second neuron. But some are free-floating. Some are unbound or free. These free neurotransmitters are constantly taken back. So you don't have a whole lot of neurotransmitters in the synapses staying long enough to bind to receptors. And then if we somehow reduce the reuptake rate, if somehow we can reduce the speed of reuptake, and then there would be more numerous neurotransmitters in the synapses. This would be like a flood of neurotransmitters, and then the receptors will get stimulated more. Then the serotonin activities, the signals will be stronger. This is how depressive medication works. Depressive medication reduce reuptake. They are reuptake inhibitors. So the serotonin binding increases with receptors. Different neurotransmitters have different receptors. Each particular kind of neurotransmitter binds only with its own receptor. Serotonin binds with the serotonin receptors. Dopamine binds with dopamine receptors. It's like a lock and key. A key opens a particular lock. You cannot open a lock if you have a wrong key. The neurotransmitter is like the key. The receptor is like the lock. Different neurotransmitters work in different parts of the brain. Of course, there's a lot of overlap. Many neurons use different kinds or types of neurotransmitters at the same time. Acetylcholine is another type of neurotransmitter with important functions. One major function of acetylcholine is muscle contraction. Acetylcholine is released from motor neurons and stimulates the muscle fibers, so the muscle contracts. Let's look at a picture. I think I have a picture showing muscle contraction. Yeah, here. This one If you look at the motor neuron, well, which one is the motor neuron? The blue line is the motor neuron. The motor neuron sends a command control information to the muscle, and it stimulates muscle fibers. And here, acetylcholine is very important, produce an important function, important role in muscle contraction. This means that if somehow you have weak acetylcholine activities, you're going to have a difficulty moving your arms and legs. You may become paralyzed. Some poisonous spiders produce a kind of a poison that weakens acetylcholine activities and make people paralyzed. There's also research showing that acetylcholine have something to do with Alzheimer's disease. Alzheimer's disease usually happens in older people, and it may start with memory malfunction, memory loss, and gradually the person loses most cognitive abilities, become demented. It's a kind of a dementia. Research shows that acetylcholine, while the neurons that use and produce acetylcholine decay or dies off in people with Alzheimer's disease. A third type of important neurotransmitter is called dopamine. Dopamine have many different functions, very important functions. One important function is that it's a reward. It's part of the human reward system. It's part of the pleasure system. Dopamine activities produce pleasure. And also, if you engage in activities that give you pleasure, usually you witness an increase in dopamine activities. Food releases, especially good food, tasty food, the food you really enjoy, gourmet food, increases dopamine production, and sex increases dopamine production. Some drugs that give people a high produce dopamine, increase dopamine production. So it's very rewarding hormone. On the other hand, dopamine are implicated in some mental diseases. In particular, schizophrenia or psychosis are associated with overactivity of a dopamine system. Well, dopamine is particularly implicated in the symptoms of schizophrenia, like hallucination. When we talk about abnormal psychology, we're going to discuss the symptoms of schizophrenia. One type of symptom is hallucination. There are different hallucinations. Particularly common type of hallucination is called auditory hallucination. You hear voices. Some people hear voices that nobody can hear. For example, some people with schizophrenia, they have they hear voices in their head that is speaking out aloud to them, is broadcasting their own thought. So a schizophrenic person thinking about something, they hear their thoughts being spoken out by a foreign voice, not their voice. Somebody is speaking out, broadcasting whatever they're thinking. And some people with schizophrenia, they hear voices like telling them to do something, giving them instructions. And this kind of symptoms are associated with increased dopamine. And delusions are also associated with dopamine activities, overactivities. Delusions are different from hallucination. Hallucination is a sensory. You hear voices, you see images. Some people with hallucination see monsters, angels, animals that are not there actually. It could be auditory, it could be visual, it could be tactile. Some people with hallucination feel that there are worms or bugs crawling all over them, but actually there's none. Delusion is different. Delusion is thinking. Delusion is mostly related with beliefs. Some people with persecutory delusion believe that there are people trying to harm them, people trying to hurt them, people trying to spread gossip about them, and they even believe some people are trying to kill them or get them killed. This is a delusion, and delusions are sometimes associated with overactivity in the dopamine system. In order to treat people with delusions, hallucinations, schizophrenia, we need to reduce the dopamine activity. And medication treating this kind of people with schizophrenia usually reduce their dopamine activity. Now, another function of dopamine is implicated. Dopamine is partly responsible for motor control, controlling motor movement. There's a particular kind of disease called Parkinson's disease. Parkinson's disease usually happens in older adults, the elderly. The main symptom is shakiness. The person cannot control his hand, feet from shaking, very shaky. So if a person with a Parkinson's disease is carrying a glass of water, his hands shake constantly. So the water would kind of be spitting out all over him. And some old people, elderly, they have their jaws shake all the time. If you observe them, it seems that their jaws are always shaking. They cannot control the shaking. This is partly due to neurons producing dopamine and use dopamine die off in the brain. So people with schizophrenia, after taking dopamine medication for years, for decades, they may develop symptoms similar to Parkinson's disease patients. Their dopamine producing neurons are damaged. A couple of key concepts about neurotransmitters: agonists and antagonists. Agonists means helpful or helper. Agonists is a molecule other than a neurotransmitter, but has a similar structure to the neurotransmitter, so it binds to the receptors. At the same time, when it binds to the receptor, it does the job. It stimulates the second neuron, and the second neuron fires. So agonist is like a booster. Agonist has the same effect or similar effects as a real thing, as a real neurotransmitter. On the other hand, antagonist has the opposite effect. An antagonist of a neurotransmitter has a similar structure with a real neurotransmitter, so it binds to the receptor, but it doesn't do the work, it doesn't do the job. It just occupies the receptor, so the real neurotransmitter cannot bind to the receptor, but the antagonist doesn't do the job of the real neurotransmitter. So overall, if you administer antagonist, then the overall neurotransmitter activities are going to be weaker. when you are treating people with schizophrenia, because the problem is partly related with overactivity in dopamine neurotransmitters, then you need to use antagonist. You need to use a dopamine antagonist to reduce the overall dopamine activity in the brains of schizophrenia patients. Okay, let's take a break now.